Everyone, and thank you for joining us for the FASEB and AAMC presentation, Animal Research Still Necessary for Understanding Human Disease. The webinar will begin in a few moments. You should have a link in your email on how to join this webinar, otherwise you would not be here. My name is Naomi Harlan-Bacchus. I am the Science Policy Analyst here at FASEB. If you have any questions, please refer them to me and I would be happy to help you. So we will get started in a few moments as we wait for the remaining folks to trickle in. Okay, everyone, it is the top of the hour, so we're going to go ahead and get started since we have a lot to cover today. Good afternoon to those of you who are just joining us. Welcome to the FASEB and AAMC webinar, Animal Research Still Necessary for Understanding Human Disease. We are privileged today to be joined by our three speakers, Dr. Cohen Van Rompuy, Dr. Nicola Mason, and Dr. Corinna Ross. And they will be speaking to us about the continued importance for animal research. So most of the time when we talk about animal research, we refer to common medical breakthroughs that come from discoveries in animals. Things like the invention of the cardiac pacemaker, the development of vaccines for measles and polio, all of which remain vital for human health. But today we're going to take a look at some more recent developments that can be attributed to animal research. Our speakers will highlight their own research and discuss the importance of selecting the appropriate animal model for their research question. So as I mentioned just a moment ago, we are joined by three speakers in three different time zones, believe it or not. Dr. Cohen Van Rompuy comes to us from UC Davis and the California Primate Research Center, Dr. Nicola Mason from the University of Pennsylvania School of Veterinary Medicine, and Dr. Corinna Ross from Texas A&M University San Antonio and the Southwest National Primate Research Center. And just to let you all know, this webinar is being recorded and it will be available on the FASEB website by about early next week. And there will also be a time for questions at the end of today's presentations. So to ask a question, please just follow the directions as you see here on your screen, and we will answer them in the order they were received. So please also know that if we do run short on time, which is probably likely, and if we have not gotten to your question, we will still collect all of the inquiries for our speakers, and we will post their answers in a Q&A document alongside the recording of the webinar. So just please rest assured your question will certainly be addressed even if we do run out of time. So without further ado, I would like to introduce our first speaker, Dr. Cohen Van Rompuy.
Yes, thank you very much for giving me the chance to talk today. So I'll be discussing how the use of non-human primates can still play a major role in vaccine development, and I'll be using Zika virus as a very nice example of this. So basically, early 2016, as you may remember, Zika virus really caught the international attention. and was among the major headlines of the news for months. In February of that year, WHO even declared it a public health emergency of international concern. Zika virus itself was not new, as it was originally discovered nearly 70 years ago. The virus was discovered in the Zika forest of Uganda, where researchers were using rhesus monkeys that were held in cages in the forest as sentinels to study yellow fever. And one day they observed that one of the animals had developed some fever, and they were able to isolate a new virus. Because of the location, they called it Zika virus. And for several decades, there were some sporadic reports describing suspected cases of Zika virus infection of humans in that region, but overall infection was considered relatively benign. However, the situation really changed the past decade when the virus slowly made its way across islands in the Pacific Ocean until it eventually reached South America, where it found a very large population that had no immunity at all, that was totally susceptible to infection. The rapid spread of Zika virus was really facilitated because it can be transmitted by mosquitoes, in particular 2 species, which are commonly found in many tropical and subtropical areas. But also other routes of transmission are important, especially as you will soon hear more about the transplacental route. For most adults who become infected with Zika virus, there are no symptoms at all. Only 1 out of 5 adults would have some symptoms, which are usually non-specific and self-limiting. However, when a woman who is pregnant becomes infected for the first time during pregnancy, there is a chance that the virus transmits across the placenta and can infect the fetus. And this can lead to a number of complications that we collectively call the congenital Zika syndrome. The most visible of this is microcephaly, in which babies are born with abnormally small brains and heads. But other complications include also fetal death and other deficits in fetal development. And currently, there are no approved vaccines or antiviral drugs to combat Zika virus. The reason Zika virus has not been that much on the news lately is that there has been a big decline in Zika cases in Central and South America, as you see here on this graph. The epidemic really peaked in 2016 and then infected the majority of the population, which led to the buildup of herd immunity. However, as this article of the New York Times of 2 weeks ago also indicates, there is no reason whatsoever to be complacent. There are many areas of the world where the mosquito vectors are present. And so the question is not, will there be another outbreak? The question is, when will this outbreak happen? We are really sitting on a ticking time bomb. Ideally, we'd like to have a Zika virus vaccine. Such a vaccine could be given to women before they would become pregnant. And so the goal of such a vaccine is to induce immune responses that, if the woman becomes pregnant, can protect both her and her fetus against Zika virus infection. The vaccine that's the furthest along in the pipeline is a Zika DNA vaccine that expresses some of the external proteins of the virus. And this vaccine was originally developed by the NIH. It was first found to be highly effective in non-pregnant monkeys in a regimen of 2 doses. It then moved successfully through a phase 1 trial and then entered in phase 2 trials about 2 years ago. However, the big question is this vaccine effective in protecting adults, but especially also the fetus? Testing the efficacy of a vaccine is really the easiest when you have an actively ongoing epidemic. However, as I just discussed, there has been a big drop in Zika cases in endemic areas. Because it's very difficult to predict where the next outbreak will happen, it's very challenging to set up a clinical trial that would use clinical or virological endpoints for adults. And it's even more difficult if one wants to use congenital Zika syndrome as the clinical endpoint. In such a context, the idea has been raised that safety and immunogenicity data from human trials, combined with efficacy data from a relevant animal model, may be sufficient to license a Zika virus vaccine. Non-human primate models of Zika virus infection are especially relevant. Non-human primates, such as macaques, are very close to humans, much more than rodents are. Several research teams, including ours, have demonstrated that we can infect macaques with contemporary Zika virus isolates, and that the infection recapitulates many of the features of infection of humans. Although so far nobody has been able to reproduce fetal macaques with microcephaly, studies have shown that it induces placental and fetal brain abnormalities and also leads to increased rates of fetal death and stillbirth. So we decided to use this animal model to test this Zika DNA vaccine. And so in short, we first immunized adult female monkeys, then we get them pregnant, and then we expose them several times to Zika virus to determine whether there was protection of the fetus. Gestation of rhesus monkeys is about 165 days, and so if you line this up with a human pregnancy, we can also divide this up in 3 trimesters of each about 55 days. So we decided to inoculate the animals 3 times, starting in the first trimester through the end of the second trimester, alternating between 2 strains of Zika virus, one from Puerto Rico and the other one from Brazil. And in our study, we had 2 arms. The vaccine arm had 13 animals that were pregnant, and the control arm had 12 animals. And so at the end of gestation, we did C-section to collect fetal tissues. Because rhesus macaques are seasonal breeders, and by the time we started the study, we were late in the breeding season, we started time-limited breedings right after the first DNA immunization. As we don't really have control on when animals become really pregnant, that means that the time interval between the second DNA immunization and the first Zika inoculation also varied. And it ranged from 4 days to approximately 1 year. And so 10 animals became pregnant quite fast, within 100 days. The last 3 animals required many time breedings to get them pregnant, from 260 days to a year. Although this means that most animals were not challenged with Zika virus at a time of peak immune responses, this larger time window really reflects a world, let's say a real world scenario. So let's briefly discuss the results. So due to the limited time, I'll only present some of the main data. While none of the adult monkeys had any symptoms, I will discuss that we had some cases of early fetal loss. I'll also be presenting the virus levels in the maternal blood, antibody responses, and then discuss what happened to the fetuses. So in the control arm, we had 2 animals that had early fetal death. When on ultrasound, we saw that there was no fetal heartbeat. So this happened on gestational days 36 and 60, so that means 6 days and 30 days after the first Zika inoculation. So we then did surgery to remove these 2 dead fetuses, and both of them had evidence of high levels of Zika virus. So we can really contribute these fetal losses to early transplacental transmission of Zika virus. In contrast, in the vaccine arm, there were no early fetal losses. We also took regularly blood samples of the mother to measure viral RNA as an indicator of virus replication. And so here I'm just showing you a very brief summary of the data. We saw that in the control arm, all animals had a very high peak of virus early on, and then most animals had prolonged detection of viral RNA. In contrast, the animals of the vaccine arm had reduced levels of virus in the blood. 8 animals had a transient detection of a small amount of virus, and 5 animals never had any detectable virus in the blood at all. It was known from earlier studies that this vaccine induces neutralizing antibodies. So we analyzed neutralizing antibody titers on the day of the first Zika virus inoculation in the vaccine arm, and we found that animals that did not have any virus in the blood had higher antibody titers than animals that had some virus in the blood. And so the cutoff titer in neutralizing antibody titers was about 1 in 300. And so what's really nice is that this is in the same range as the titer that had also been observed in human trials using the very same assay. So this really indicates that the titer of neutralizing antibodies in the blood is a very useful immune correlate of virus replication in the mothers. Of course, the central question is really, did this vaccine protect the fetus? So therefore, we analyzed a long tissue of fetal tissues to look for viral RNA. And so this list includes tissues of the maternal fetal interface, the fetal brain, and also other fetal tissues. The next slide here shows the heat map of viral RNA. So the heat map basically means that the darker the color, the more virus was in each tissue. We used as a working definition that if a fetus had at least 1 tissue that had detectable viral RNA, we consider the fetus infected with Zika virus. So on the left side here, you see the 12 control fetuses, and as you see, there is a lot of color. 11 of the 12 control fetuses had at least one tissue with detectable Zika virus RNA. In contrast, on the right side, you see the vaccine arm, and here you don't see any color at all. And that's because we could not detect any virus in any of these tissues. So this really means that according to our definition, none of the vaccine arm animals became infected. So this vaccine really worked tremendously in reducing viral infection rates. None of the fetuses had evidence of microcephaly. We had 2 pathologists who were blinded to the animal assignment, and they looked at many fetal tissues, and extra attention was given especially to the fetal brain, the placenta, and also the amniotic membrane. And they developed a scoring system that ranged from normal to severe. And so a low score of 1, which is minimal, is really still of unclear clinical significance because the many procedures, including sedations, can also lead to some minor changes. And so while I don't really have the time to show the full details, I can state that the vaccine reduced the pathology scores. The control arm had a significantly higher pathology score than the animals of the vaccine arm. Here in the control arm, we saw that the brain lesions we found were really consistent with what we had seen in other Zika studies, including loss of the epidermal cell line, and also some evidence of brain gliosis. And we also saw the typical Zika-induced lesions in the placenta, including infiltrations of many neutrophils and also necrosis. So in conclusion, this study in a relevant animal model really shows how a model can really be very useful to help to advance a certain vaccine. Here we showed efficacy of a DNA vaccine, and in the course of this study, we got a number of important lessons that can really guide and complement clinical trials, and that hopefully can lead to approval of a Zika virus vaccine in the next few years. So in particular, we saw that protection against prolonged maternal viremia can be a very useful surrogate marker of fetal protection. We saw that neutralizing antibodies correlate with viremia. We saw that protection was durable for at least a year after the vaccine. And we also saw that sterilizing immunity of the mother was not required to have protection of the fetus. And last but not least, I really want to thank the many collaborators who worked on this study. It really takes a village to make these studies happen. So thank you very much for your attention.
Thank you so much, Dr. Van Rompuy. That was fascinating. I think the most striking was the table that showed the control arm and the vaccine arm and how much virus was in each of those treatment groups. And it's just striking that vaccines had absolutely none detected. It's just incredible. I have one follow-up question. So you mentioned early on that non-human primates are genetically similar to human, more so than rodents are. But what about rabbits and other members of the hare family? Why is it not possible to study Zika in a species like a rabbit?
Yeah, so that's actually a very good question. People have tried to infect rabbits with Zika virus and have not seen any evidence that the virus can replicate in rabbits. So that's why to study, let's say, efficacy of vaccines or other interventions, we really have to use a host in which the virus can replicate.
Great, thank you. Okay, we will move on to our next presentation given by Dr. Nicola Mason. Please take it away.
Yes, thank you very much, and thank you FASEB, and thank you to the AAMC for the invitation. So I want to take this time just to talk a little bit about the development of a vaccine that delays metastatic disease and prolongs survival in patients that develop a cancer called osteosarcoma, a bone cancer. Next slide, please. Now, why are we actually looking at this? Why are we looking at developing vaccines? I think when we actually look at the average cost to develop a new drug for human use here, you can see in this table, if you look at the right-hand side, the total cost is about $2.5 billion to develop a new drug for human patients. And the reason for this is that many drugs that actually move into the development phases actually fail. Next slide, please. On this next slide, you can actually see the likelihood of approval from phase 1. And you can see that particularly with respect to cancer drugs, the likelihood of approval is extremely low, so 5%. So 5% of drugs that enter into phase 1 onwards clinical trials in humans are approved, meaning 95% actually fail. And the main reason that they fail is because they're either found not to be safe or they're found to have lack of efficacy. Next slide, please. So what this really means is that the way that we are selecting drugs to go into phased clinical trials in humans clearly is flawed, because 95% of them are failing. So the preclinical models that are used, shown here in the top right-hand corner, the mouse and then the beagle and non-human primates, are obviously not accurately predicting the safety and efficacy of these drugs. So what we are suggesting is that if we integrate another parallel patient population into this drug development paradigm, this may actually help us, particularly in the preclinical phase, to identify safer and therapeutically effective drugs that can then move through into the human clinical trials. So what is it about dogs then that make them popular, as it were, to actually get integrated into this drug development paradigm? Well, there's a lot of them. There's 84 million of them, and 4 million of those in the United States will go on and develop cancer. And the cancers that you can see are listed there. In particular, relevant today is osteosarcoma. So again, just to emphasize, these are pet dogs. They're dogs that live in your household or my household that spontaneously naturally develop the disease. And when we look at those types of cancers in the dogs, they are very comparable with respect to their biology, their behavior, and their genetics. When we treat dogs in the clinic with chemotherapy or surgery for cancer, we recognize that their cancers will come back, they will recur, they become chemo-resistant, and they suffer from distant metastases, all problems that we see in humans. And particularly when we're looking at immuno-oncology drugs, so using the immune system to target cancers, this is very important that the model, if you like, that we are using to evaluate them is immune competent. So in other words, has an intact immune system which has already edited and sculpted the tumor. And this is very important. And again, in our pet dogs, they have intact immune systems. And again, immune defects that we see in humans are also recapitulated in the dogs. Next slide, please. We also know that the dogs are treated, as you can see in the bottom right-hand corner here, with the same types of drugs or radiation or surgery as humans will be. So that gives us a very comparable background in which to study these drugs in combination, and particularly in patients that have had treatments that might affect their immune system. Next slide, please. So I really want to just emphasize the value of dogs in research here by taking the case of osteosarcoma. So osteosarcoma is a very aggressive tumor that occurs naturally in children and also naturally in dogs. And you can see on the left-hand side, this is what osteosarcoma looks like in a child. Proximal humeral bone lesion, large swelling, painful swelling, and you can see beneath that is an osteosarcoma that's occurred in the distal radius of this greyhound. Radiographically, we see features of lysis, bony lysis, and bony proliferation, this very classic sunburst appearance. And the treatment of choice, both in children and in dogs, is to remove the primary tumor. So you can see in both of these cases, the child and the dog has undergone amputation, or perhaps now, at least in children, more commonly limb-sparing surgery. But in about 30% of these children, they will go on to develop metastatic disease in their chest, and there is nothing that we have that can effectively treat these children. In over 90% of dogs, they will go on to develop metastatic disease in their chest, and we as veterinarians have nothing to actually treat that. So really what we need is a new agent that can prevent metastatic disease in both children and dogs. Next slide, please. So we do actually know that osteosarcoma is immune responsive, and this is work from William Coley back in the late 1800s. He was a bone surgeon, and he recognized that patients coming into his clinic that had bone sarcoma, that had concurrent bacterial infections, tended to do much better. And the thought was that those bacteria were in some way non-specifically stimulating the immune response such that it could go and find tumor cells around the body and eliminate them leading to longer survival times. And we have seen from the literature cited here exactly the same thing in dogs, that in canine patients that have had limb salvage surgery for osteosarcoma that develop bacterial infections, they survive about twice as long, which is about 2 years, versus the 1 year of survival of dogs that have standard of care amputation and chemotherapy. And on the right here, some very elegant data using comparative transcriptomics, demonstrating that human patients, as well as canine patients that have a lot of immune activity within their tumors, tend to survive longer. Next slide, please. So this gives us an idea that stimulating the immune system might actually be a way of treating both dogs and humans with osteosarcoma. But could we direct the immune system against a specific molecule, a tumor-associated antigen? And we are looking, and others are looking at HER2/neu, an epidermal growth factor receptor. And we and others have found that this is expressed in both canine osteosarcoma and pediatric osteosarcoma. So could we develop a vaccine that targets HER2 to prevent metastatic disease? Now, it's difficult to do that trial in children because there's only about 400 children that will develop osteosarcoma per year, but there's about 40,000 pet dogs that will develop osteosarcoma. So we have essentially a spontaneous model in which we can evaluate the use of a HER2 targeted vaccine in preventing metastatic disease after standard of care. Next slide, please. So we actually went on to look at the literature, and you can see here these are Kaplan-Meier survival curves demonstrating that pretty much over the last 30 years, when people have used combination chemotherapies, these are dogs that have osteosarcoma, and pretty much doesn't matter what you do, these dogs usually die with a median survival time of around about 320 days. Next slide, please. So we designed a listeria vaccine, a bacterial vaccine that had been genetically modified, one, to be highly attenuated so it doesn't make the patient sick, and secondly, to actually contain that target molecule HER2/neu. And that live attenuated bacteria is delivered to the patient intravenously, is taken up by these mononuclear cells, these antigen presenting cells, and will actually present antigen in the context of both MHC class 1 and class 2 to stimulate both CD8 and CD4 T cell immunity respectively. Next slide, please. So we went on to develop a phase 1 clinical trial in pet dogs with spontaneous osteosarcoma that came into the clinic, were diagnosed with osteosarcoma. They would have the tumor removed, that's standard of care, screened to see if they express HER2/neu, and then they'd have follow-up chemotherapy, which is also standard of care, before being screened to make sure they have no evidence of metastatic disease at that time point, and then receiving 3 doses of this HER2-targeted listeria vaccine, and then following the dogs out every 2 months to see when they developed metastatic disease. And you can see in the table the types of dogs we had, and we were actually doing a dose escalation to look at safety as well as efficacy. Next slide, please. So these were actually the responses. So these are Kaplan-Meier survival curves looking at the survival of the vaccine group versus a historical control group. And we actually found a statistically significant improvement in survival in those dogs that had received the vaccine versus those that had not. And if you look at the table underneath, you can see that the median survival time over doubled in terms of the vaccinated group, with 956 days compared to the historical group of 423. So that's almost 80% of our patients surviving 1 year, compared to half of them with the historical control, and almost 70% of them surviving 2 years, compared to 28%. And this vaccine now, through this work, has received a conditional license to be used in veterinary species, in dogs with osteosarcoma, and we are working closely with the Children's Oncology Group based on this data to try to get this now into pediatric trials. Next slide, please. So the other thing that's useful in the dogs is looking at correlative biomarkers, and one very simple thing we did here in this clinical trial was to look at the white blood cell count and the neutrophil count 24 hours after vaccination. And what we found is those dogs that had a higher increase in white blood cells and neutrophils in their peripheral blood 24 hours after vaccine survived longer than those that didn't have such a great increase in neutrophils or white blood cells. So that might suggest to us that's a very early potential biomarker in which we could use to predict who might respond to this vaccine and who might not. Next slide, please. This is a complicated table, and I don't have a lot of time to go through it, but really what I want to point out is the column on the left. We are looking at all the dogs and we're looking at their baseline responses against HER2/neu. And you can see that only one of those dogs had some response against HER2/neu. None of the other dogs had evidence of immune response against this molecule. But after vaccine, as you go through the different columns, you can see that the plus signs indicate the responses in an interferon gamma ELISpot assay to HER2/neu. And you can see that a number of these dogs developed these responses, indicating that the vaccine was stimulating HER2-specific immunity. And on the right-hand side, you can see the overall survival of these dogs. Remember, classically, it's around about 300-400 days, and you can see a large number of these dogs going out over 1,000 days, with a number of these dogs actually still alive. Next slide, please. So we think the mechanism of action here is that the listeria is inducing T-cell responses, both CD4 and CD8 T-cell responses, to kill remaining tumor cells after amputation and chemotherapy, and also is stimulating the innate immune system to produce IL-12, which can upregulate FAS on the surface of tumor cells. So when they actually pass through the lungs and try and settle there to cause metastatic disease, they are eliminated through the constitutive expression of FAS ligand in the lungs. And actually on the right, what you can see, this is actually a patient who received the vaccine who had pre-existing metastatic disease. And you can see here we did minimally invasive surgery to remove one of the lung lesions that that patient had. And you can see all the T cells as indicated by CD3 on immunohistochemistry lining up here around that metastatic lesion, trying to get in and trying to eliminate that tumor. Next slide, please. But what is quite evident from that immunohistochemical slide is that the T cells weren't getting into the tumor. This is a classic example of T cell exclusion, which we see in human patients being treated with a number of different immunotherapies. And this then leads us on to say, well, perhaps if we were to combine this therapy with checkpoint inhibition, which basically essentially releases the brakes on the tumor and releases the brakes on the T cells to allow the T cells to actually act on the tumor. This could actually be an effective way, perhaps, to evaluate this type of approach in patients, both canines and children, that have pre-existing metastatic disease. Next slide, please. So in summary, osteosarcoma is an immune responsive tumor. We think that targeted immunotherapies hold a lot of promise for delaying or even perhaps preventing metastatic disease when it's administered in the setting of minimal residual disease, both for dogs as well as for humans. I haven't had time to show you combination data using palliative radiation with immunotherapy. But this is something that we are looking at again in canine patients to broaden that anti-tumor immune response, perhaps prior to a definitive surgery. And it's really, I think, quite exciting to know that these immunotherapeutic options now are available perhaps to effectively treat or perhaps, dare we say, cure osteosarcoma in the veterinary space. And now, of course, we need to develop rational and hypothesis-driven, mechanism-based, prospectively designed clinical trials that will actually answer the best way to utilize this vaccine and then move it into the pediatric space and hopefully bring an effective treatment to children that are suffering from this disease. Next slide, please. So a lot of people that are involved in these sorts of studies in the lab as well as in our veterinary hospital, all of whom I'm grateful to for their assistance and help, and of course our funding agencies as well. Thanks very much.
Thank you so much, Dr. Mason, and I just find it so heartening to see that your research is actually not only helping potentially people and children with this disease, but also the dogs that have this disease too, and so many of them will eventually develop. And so just to kind of take a step back and take our scientific hat off for a moment, when you say spontaneous, you mean that the dogs already naturally have the disease and you are not effectively giving them this cancer?
No, that's exactly correct. These are patients that walk into our veterinary hospital here at the University of Pennsylvania because the owners have noticed that they are limping, they're lame, and we diagnose them then with osteosarcoma through our standard diagnostic tests. So that's exactly right. And rather perhaps than using the word model here, the better perhaps approach would be to say this is a parallel patient population that also is in need of new effective therapies. So we can essentially kill two birds with one stone. In other words, developing therapies that will benefit people's pets as well as children.
Exactly. That's so fascinating. Thank you so much. Okay, so we will continue on with our last but not least presentation by Dr. Corinna Ross.
Thank you. I'd like to thank the organizers for allowing me to participate in such an amazing webinar. Next slide, please. I'd like to start by introducing marmosets as an animal model, primarily because I know a lot of people aren't as familiar with marmosets. Marmosets and tamarins are primates that belong to the family Callitrichidae. They're New World primates found natively in South America. And in the 40s, 50s, and 60s, these animals were imported into the US, Europe, and Japan for biomedical research and for populations in zoos and other captive situations. And a lot of these animals were used for biomedical research and then have been phased out. So what we have remaining is the species on the bottom right, the common marmoset, the little guy with the bright white tufts, that's primarily found in biomedical research. Next slide, please. So these animals offer a lot of unique adaptations and unique benefits for research. They are small animals. There are about 350 to 450 grams. So to put it into everyday comparison, they're about squirrel sized. So they're fairly easy to house and to take care of. And they mature very rapidly due to the small size. So typically marmosets become sexually mature at about 1.5 years. Their average life is about 6 years, and those that live beyond 6 years, we consider them old age at 10. They start to show some geriatric signs, and the maximum lifespan ever reported in captivity is 22 years from a marmoset kept in Japan. Next slide, please. So amongst primates, marmosets have a number of really unique characteristics besides being small. They're one of the few non-human primates that gives birth to litters. We used to think of them as being obligate fraternal twinners, so that the females give birth to twins on a regular basis. But what we're finding is that it's actually quite variable, and the litters can range in size from 1 to 5. But twins and triplets are the most common. And in this primate, they're not seasonal breeders. They'll breed throughout the year, and so they will produce as many as 2 litters per year because they have a fertile postpartum estrus. So females give birth to twins, and then in approximately 2 weeks, they can become pregnant with their next litter, which means that we can study a lot of questions regarding reproduction, and they produce a number of infants in a short period of time. This energetic need, we think, drives a number of their social adaptations. So as a primate, they form socially small family groups where it's a male and female breeding pair, their offspring, and then the older offspring stay in the family group to help take care of the younger babies and get experience with those younger infants. And this is cooperative infant care. Uniquely, males are the primary caregivers. So while the females are lactating and providing that care and gestating the next set of twins, the males become the primary caregiver. So this image is of a male with his 2 young infants. Next slide, please. So marmosets have played a major role in biomedical research, as I said, since about the 40s, and they've been studied for a number of things. A lot of work has been done to define that social network and the social family and what happens in that dynamic, because it so closely mirrors what is the typical human social situation. And so a lot of behavioral endocrinology research was done. We've done a lot of work in reproduction, including the production of transgenic marmosets, which are pictured here on the right. And marmosets were one of the first primates not only to become transgenic, but to show germline transmission of these transgenes. We can also, because of their rapid reproduction, study questions like developmental programming. So what happens in the womb and how that might affect later health. a lot of work in immunology, neuroscience, and they're also one of the few primates that develops pediatric obesity. So there's a lot of different things with the marmosets that have been very useful to help us model human diseases. Next slide, please. We're going to focus today, I'm going to focus on aging and our work in aging. And people might wonder why we focus on aging, and this is primarily because we're all aging. And even if we don't want to admit it, it's the primary risk factor for a number of diseases. So Alzheimer's, Parkinson's, cardiovascular disease, cancers, all of those, the primary risk factor is the fact that we get older every single day. And Americans are fascinated with trying to prevent aging, often with completely useless treatments such as lotions and antioxidants and laser therapies, none of which are slowing the aging process truly. But what we notice is that we all know individuals that show variability in their aging. We know individuals that are 60, 65 and they look like they're 40. And we know other individuals that are 40 and look like they're 70. So understanding that variability and what causes people to age in different ways and have different health outcomes is of real interest. And our lab isn't interested in extending human lifespan. We're not looking to have a lot of people that are living to be 130 and living in nursing homes. Instead, what we're interested in helping individuals that are aging to 80 and 90 be independent and have lower risk for those diseases and outcomes. Next slide, please. So in order to do this, we've been developing for the last decade or so the Marmoset as a model for aging questions. And the reason for this is I told you already that they have a shorter developmental time frame. And that can be really important if we're interested in interventions that can change how you age from midlife to old age. If we try to study that in humans, it's longer than the lifespan of the investigator. And we can use other non-human primates like the rhesus and the baboon, but they live a long time as well. So it extends the length of those studies. Whereas for the marmoset, we can go from midlife at 5 to 6 years of age into their geriatric frame in a 5 to 10 year time span and get a lot of information about the effect of these interventions. We can do similar studies in rodents, and people do. Rodents are a primary model for aging, but a lot of the physiological outcomes for rodents are different than what they are for non-human primates. They have different nutrition, they have different metabolic physiology, they have different sleep patterns. So to truly convert it into a preclinical model, we need a non-human primate. Next slide, please. So to first characterize marmosets and aging, we were first interested in whether marmosets show the same causes of death or similar causes of death of what we see in aging humans. And what we find is that animals over that age of 6 going into the geriatric years show increased rates of cardiac failure, increased rates of kidney disease, amyloid deposition, those types of things. And so it very much mirrors what we see in humans. Next slide. And so we focused on looking at domains of healthy aging, and specifically 5 domains of healthy aging. And these are domains that geriatricians look at when individuals come into their clinic. So they will rate them on how they are regulating their physiology, their immune responses, their metabolic status, mobility, and cognition. And they usually have a battery of tests that they use to assess individuals to see what kind of risk they are for frailty, Alzheimer's, those types of things. So we've been translating those measures into the marmosets to come up with tools to assess healthy aging in the marmoset. And due to today's time constraints, I'm going to just focus on 3 of these levels to show you some of what we see with the marmoset. Next slide, please. So in homeostatic aging, what we're looking at is how an animal regulates its health and regulates its physiology. And so one of the good markers for human aging is blood pressure. We all know that as people age, they end up with higher blood pressure. They're at a higher risk of having to be on medication to control that blood pressure. And what we see is when we look at geriatric marmosets, we see the same thing. We see a dysregulation of blood pressure and higher arterial pressure as they age. Another marker that's of new interest is the microbiome. And these are microbial organisms that live in the gut, and we're starting to learn just what that microbiome diversity does for health. It's been implicated in obesity in humans, it's been implicated in mental health in humans, so we're still trying to tease out what it does. And one of the reports is that microbiome diversity decreases with age in humans, but these humans are conflicted with being in nursing homes, being on antibiotics, being socially isolated. So we wanted to test it in an animal that we have control of the environment and know what's happening with them. And we did this in the marmosets. And we see that elderly marmosets have decreased microbial diversity, even though they have the same nutrition, they're in the same social system, and they are not on antibiotics or other medications. Next slide, please. When we look at mobility, mobility is one of the markers in humans that we see as high risk. Older individuals are much more likely to fall and risk breaking a hip or other bones. So we wanted to look at the marmosets, and what we see is a decrease in activity as they age, but they maintain normal social behaviors. They're still eating together, they're still grooming together and socially interacting. And what you see in this image is the marmosets are wearing little backpacks with Fitbit-like devices. So we're able to count their steps throughout the day and see how active they are and how often they're just at rest. One of the things that we found is an interesting stretching behavior that's significantly associated with risk of death in the next 6 months when they start expressing it at a higher level. So this is a behavior that we can look out for to decide on animals that might be at higher risk for bad health in the future and bad health outcomes. So if we can find these types of biomarkers, we might be able to start predicting things like frailty in the humans. Next slide, please. One of the main reasons to use a non-human primate model for aging is to be able to look at cognition. Primates have a different type of cognition and brain physiology than other mammals. And so this is my favorite task. We've been working on several to assess cognition, but this is the conveyor belt task. And basically from a human perspective, picture I Love Lucy, the episode where the candies are coming down the conveyor and she's trying to rapidly pick up as many as she can. So in this task, what happens for the marmoset is they're presented with multiple treats. The first one is an apple, and apples are okay, but they don't want the apple. They want the marshmallow because they love marshmallows. So they need to have control of executive function and impulse control in order to wait for the first treat to go by and then grab the second treat. So young animals rapidly learn, they make a mistake the first time, and then they rapidly learn to wait for the marshmallow. Older animals have an impairment in that learning task, and they will continue to grab the apple. They eventually learn to wait and get the marshmallow, but initially they have a learning impairment. So we're able to assess that cognition in aging. Next slide, please. We're also able to test preclinical interventions, and this is really what we want with the marmosets, is to be able to take things that we hypothesize will increase healthy aging for humans and test it in the marmosets that have the shorter lifespan to see if it not only helps with lifespan, but with health. And rapamycin is a drug that's already FDA approved for use in humans. It's used for organ transplant and for cancer treatment. And it was tested by the Intervention Testing Center in mice to see what the effect might be on aging. And it turns out that it extends lifespan in mice models, and it also extends health. So rather than, though, giving it straight to humans with there's possible side effects, and we don't want to give geriatric, healthy aging geriatrics a disease or symptoms, we decided to bring it into the marmoset as a preclinical model, and it's currently being evaluated in a longitudinal study. So far, it appears to be protecting kidney function and cognitive function. So we're still in testing, but we hope to have some conclusions soon for that. Next slide, please. So in conclusion, marmosets make a good model for a number of biomedical examinations, but particularly aging. We're able to, with the marmosets, not just assess and evaluate longevity, but to really assess health that's important for translation to humans. So frailty and cognitive disease and those types of things that are higher risk for humans. And marmosets allow us to translate things that we can do with mouse models into a non-human primate model, but also in reverse. We're able to take tests that we use with humans and questions that we have with the humans that we can't control because we can't control what they eat or sleep or what they're doing, and bring that back into a non-human primate model. And we're beginning to test these interventions to see if we can promote healthy aging and increase health for humans. Next slide, please. In order to do this work, just as everyone else mentioned, I have an army of collaborators who are really great colleagues, and I'd like to acknowledge their help and the funding. Thank you.
Thank you so much, Dr. Ross. I'm still just fascinated by the fact that marmosets are the size of a squirrel. I always imagine them to be something, you know, like the other non-human primates, but that's crazy that they're that small. I had one follow-up question, I think because it's very important. What regulations are in place, both at the federal and institutional level, that ensure animal welfare is prioritized throughout all of your experiments?
That's a great question. And we spend a lot of time handling animal welfare and being aware of animal welfare. And there's a number of regulations and policies and laws put into place. So starting at the federal level, there's a number. There's the Animal Welfare Act, which guides policy by the Public Health Service and guides policy and regulations by the USDA. Most institutions that have non-human primates are also ALAC accredited, so that's the Assessment and Accreditation of Lab Animal Care group, and we also get inspected by the USDA. And all of those rules, regulations, and policies guide our institutional IACUCs, so that's the Institutional Animal Care and Use Committee. So everything that we want to do with the animals, those proposals have to go through this committee that's made up of vets and other scientists and community members to review for the scientific justification and for the potential benefits and for the impact on animal welfare. And so all of those things guide what investigators do, what animal care staff does, and what the veterinarians do.
Great. Okay, that makes a lot of sense. So animal welfare is always the #1 priority here, and I really appreciate everyone's time and presentations. And I think now we are going to move forward and take questions from the audience. Again, as you see on your screen, in order to ask a question, just please type it in the white box and hit send, and I will receive them as they come in. So I will go ahead and begin questions. So first one is for Dr. Rompe. Dr. Rompe mentions that microcephaly has not been shown in laboratory studies. The congenital Zika virus infection as a silent pathology with loss of neurogenic output in the fetal brain, which I believe is a Nature article he's referring to, describes loss of fetal, sorry, I'm working with my computer, describes loss of fetal non-cortical brain volume and other microcephaly lesions in non-human primates. So I'm guessing he's asking you to address that journal article?
Yeah, so basically, so that was a study done by, say another research group, and of course they actually saw that looking at the brain itself, they saw major changes, including also say like brain volume. But so far, based on what I know, if you look at the skull size, which is also, of course, what we would use in human babies. Say microcephaly is actually defined by measuring the circumference of the skull. So far, nobody has really been able to see the clear microcephaly, similar to what we see in Zika exposed human babies.
I see. Thank you. Our next question is for Dr. Mason. Are there other animals that get osteosarcoma naturally? Are you implicating that the use of dogs needs to be needed for more preclinical trials for other diseases for allowing it to be safe for clinical trials for humans?
Yeah, so osteosarcoma, certainly we do see it in other species. We see it, but it's much less common. So for example, we will see osteosarcoma in the cat, for example. But it seems to be particularly common in dogs, and it seems to be particularly common in large or giant breed dogs, which is interesting. One of the differences between canine and pediatric osteosarcoma is that in the dogs, it tends to happen in middle to older age dogs. And of course, in the human world, it happens in pediatrics. But otherwise, it really is incredibly similar to the osteosarcoma that occurs in pediatric patients. So that really is the key reason why we use dogs. So as I said, 40,000 or so will develop, and that's a rough estimate, will develop osteosarcoma. So that's why we use them. And the second part is, yes, absolutely. I mean, there are a number of cancers, and there are other diseases as well, that pet dogs will spontaneously, naturally develop that actually do very closely mimic the types of cancers that we see in humans. And there are a number of examples of this. Diffuse large B-cell lymphoma is one of them. Melanoma is another one of them. So there are differences, don't get me wrong. It's not absolutely identical. You need to understand the model, you need to understand the similarities, you need to understand the differences, and you need to select appropriately. But in terms of looking at safety and efficacy, yes, we think that the dogs are highly relevant for certain cancers, but also in considering the design of human trials and the ways in which these drugs are used or combined is extremely important. And again, each time we do this, we add additional information on board for our veterinary species. And as you know, I'm a veterinarian, and you know, that's our primary interest, is actually bringing new treatments to pets that don't have other options.
Great, and I think you answered our next question, but I will go ahead and ask it. Why do you think that the data on dogs will help better predict results on humans than other animal models?
Yes, well, I think that is obviously remains to be seen. It's a great question, and it really does remain to be seen. But I think that, you know, when we look at the diseases, we look at the course, we look at the biology, we look at the genetics, we look at the treatments. All of these are extremely similar between dogs and humans, and certainly much more similar than you know, any model that would be generated in a mouse compared to a human. So I think that you know, we really need to still show that dogs are predictive, but I don't think that's the only question here. Is the dog predictive? I think the dog can direct human clinical trial design, and that's important to understand as well.
Right, I think that's an important distinction, the distinction between predicting and directing our scientific experiments.
Yes.
So our next question is for Dr. Ross. What are the health effects on consistent impregnations?
Oh, that's a great question, and actually one that we're continuing to investigate. So marmosets do not display menopause, and so they could theoretically reproduce their entire lives. Most colony management controls the production after a certain age, so they will do things like birth control, vasectomize the males, that type of thing, so that the female is not continuing to produce her entire life. There is some research going into looking at bone density and the effects of estrogen and that increased rate of progesterone over pregnancies, to look at that effect long-term on females. And I think from an aging perspective, it's a really nice thing to think about looking at for humans, because we have a lot of variability in human females, some that don't ever reproduce, some that reproduce after years on birth control, or some that reproduce 8 or 9 or 10 infants. And we don't know what those long-term, I mean, we can imagine as mothers, we can imagine what that impact is, but we don't know what the health outcomes might be for those individuals long-term.
Right. Okay. And then I guess as a follow-up to that, we have a question about how translatable have past research findings, specifically treatments on marmosets, been to humans?
There have been a number of things that have translated from marmosets. So a number of vaccines have translated from development in the marmoset straight to humans and have been effective. Other things, management of reproduction, those types of things have been tested in the marmoset as well and help to guide human research. A lot of times it'll go into preclinical trials and then come back to the marmoset for retesting and go back out to humans. But examples on immunology, neuroscience, and infectious disease, those things have translated to humans.
Gotcha. And one more for Dr. Ross. What are the costs of marmoset research compared to other non-human primates?
That's a great question. So cost comes into question a lot. Is important for investigators to think about, and marmosets are significantly less expensive to maintain than are the larger primates because they're smaller, because their housing differs, and because we can house them socially the majority of their lives. So they stay in these social groups. So that means we're not having to have extra cages or space to separate them out. So they cost significantly less, but significantly more than rodents. So there's a balance there. And I always suggest to investigators that ask about moving into marmoset research that the model has to fit the question. And so we don't just use marmosets because they're cheaper. Some questions have to be asked in baboons and macaques because marmosets just aren't a good model for that. Alternatively, others should be asked in the rodent. So it's that cost balance analysis that's really needed.
Great, thank you so much. We got a comment from thanking all the speakers for answering all of these questions. We will continue on. I think we have another one related to marmoset research. Is studying these animals to geriatric age a 5+ year-long endeavor? Do you have consecutive generations every year? How does your experimental design actually work?
That's a great question. So we have some studies that are longitudinal, and we're studying them for 5 years. We have data since they were born in the colony, so it's actually much longer than that. So our animals that are 16 and 17 years old, we have all of their clinical history and their parents' clinical history. So it depends on the intervention. So there are some interventions that we're testing longitudinally, and we follow the animals for 5 or 6 years or longer. There's other questions that we can do more short term. So there's been other drugs like metformin, for example, that's approved for FDA and may have an effect on longevity and health, certainly has an effect on diabetes and obesity. So what that might be a shorter term question to look at specific physiology outcomes like glucose metabolism. And we might be able to study that in a much shorter time. But our developmental programming questions, we were able to study 3 generations of birth of a mother, birth of a female, to her own reproductive maturity, to her infant's reproductive maturity in the course of a 5-year study. So it offers a lot of advantages.
Gotcha, that makes sense. Thank you. I just came up, thought of this just now for Dr. Rompe. I saw that Dr. Ross had mentioned in her list of biomedical areas the marmoset is useful for that Zika has been recently used for research. So what are the advantages of using the rhesus macaques for studying Zika over the marmoset?
Yeah, so of course, say people have developed both the rhesus macaque model and also the marmoset model. So I think both of these models have their advantages and their disadvantages. Of course, I've only worked in a rhesus monkey model. One of the advantages that we have with rhesus monkeys is that we can, of course, since the rhesus monkeys are a lot larger, we can collect blood samples a lot more. So we actually have more plasma. We can also look at like more, say, different parameters in the blood. An advantage, of course, of marmosets is since they have multiple fetuses, is of course that you could actually also see, for example, what's the rate of transmission within a single female? Yeah? So I think both models have their value. So I think it really depends on selecting the right model for the specific research question you want to answer.
Yes, the importance of picking the right model for your research question. I think we will, for the sake of time, just take one more question and then we will wrap up because I know everyone has busy schedules. So last question for Dr. Ross, are there other behavioral assessments that can be performed in marmosets that can be translated to human conditions associated with aging?
Absolutely. So we're spending a great deal of time now actually taking computer automated testing, which they use quite a bit for humans, and translating those tasks to the marmosets. So we can't do things like ask them what time of day it is because they don't care, but we can ask them on a computer what symbols match, and that's a task that's very similar to what happens to humans. So we're working on that translation, and that goes both ways. We're taking it from human trials to marmosets and from the rodent trials and mixing them together.
Fascinating. Okay. Thank you all so much. We are getting several comments saying thank you for your time and to do this and speaking about your research. And we really appreciate you speaking on these topics that are just so important this day and age. So I guess we will wrap up now. Again, as a reminder, this webinar is being recorded and will be posted on the FASEB website along with the question and answers from each of you. For those of you that we did not get to, we will be sure to submit those questions to the speakers and have that posted as well. If you have any questions, please feel free to reach out to me. My name is Naomi Harlan-Bacchus here at FASEB. I am here to answer any questions you have related to animal research. I appreciate everyone's time and hope you have a wonderful day. Bye.